Welcome to the Brain and Behavior Research Foundation's Meet the Scientists monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation. Today, Dr. Matthew State will present Autism and Genes. The Brain and Behavior Research Foundation funds research around the world that identifies the causes, improves treatments, and will ultimately result in preventative techniques and cures for mental illness. Since 1987, the Foundation has awarded more than $324 million in research grants. We are the largest funder of mental health grants outside of the federal government. And what makes us unique is that 100% of all donor contributions for research are given directly to our researchers who are working to find the scientific breakthroughs in brain and behavior disorders such as ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, OCD, PTSD, and schizophrenia. I'm delighted to introduce Dr. Matthew State. Dr. State is a child psychiatrist and human geneticist studying childhood neuropsychiatric disorders including autism spectrum disorder, Tourette syndrome, and childhood onset schizophrenia. His lab focuses on gene discovery as a launching point for efforts to illuminate the biology of these conditions and to develop novel and more effective therapies to treat them. Dr. State co-leads several international genomics collaborations, including the NIH-funded Autism Sequencing Consortium, and he has been the recipient of multiple honors and awards, including his recent introduction into the Institute of Medicine. In 2012, Dr. State was awarded the Brain and Behavior Research Foundation's Ruane Prize for Outstanding Achievement in Child and Adolescent Psychiatric Research. Dr. State received his MD from Stanford University and earned a PhD in genetics from Yale. He was on the faculty at Yale from 2001 to 2013 and is currently the Oberndorf Family Distinguished Professor and Chair of Psychiatry at the University of California in San Francisco and Director of the Langley Porter Psychiatric Institute and Hospital. Today's webinar will start with Dr. State's presentation. This will be followed by questions and answers. To submit your questions, please use the Questions tab on the control panel on your screen. You can submit your questions throughout the presentation. Following the presentation, I will present your questions to Dr. State, and he will address as many as possible. And now, I'm pleased to present Dr. Matthew State. Matthew, the stage is yours.
Thank you so much, Jeff. I apologize. I woke up this morning with a bit of a cold, but I will do my best to be intelligible. So, yes.
Perfect. Yes, perfect.
Great. Good morning, everyone, or good afternoon if you're on the East Coast. I do want to talk today about autism and genes, and really the main point of the talk that I hope I'll leave you with is that there really has been a tremendous revolution in our ability to identify the genetic contributors to autism, and that's now setting the stage, I think, for a major transformation in our understanding of pathophysiological mechanisms. I think the key is that gene discovery, which I'm going to talk to you about, is not an end to itself, but in fact really just the beginning of a road to understand what's going wrong in brain development in conditions like autism or schizophrenia, and to use that knowledge then to begin to identify novel avenues for treatment. So I want to start today just by giving you a brief overview. See if, there we go. Yeah, it's getting ahead of me, but I will come back. Autism, the diagnosis of autism, like other psychiatric conditions, is a clinical diagnosis. It's based on observation of the patient as well as information provided, particularly in autism because it's often in young children, information provided by their caregivers or parents. The defining characteristics are a fundamental impairment in reciprocal social interaction and communication, and then highly restricted or repetitive behaviors. And I'm going to show you a bit of a video later to try to give you a sense about sort of those core features of autism. There are a number of other of clinical phenomena that often go along with autism, including, in some children, profound difficulties with language, but also other somatic or physical difficulties that go along with this condition. But the defining features are really this a fundamental problem with social interaction and social communication, as well as these unusual behaviors. It affects about 1 to 2% of the population. And typically we see the emergence very early in life, in the first months of life, and it is typically a lifelong condition, not exclusively, but typically. We've known for years that it runs in families, and it runs in families in a way that allow us to infer that that is a consequence of genetics as opposed to shared environment. There's no question that there's also an environmental role, but genes at this point are estimated to play the lion's share in risk. And I'm going to talk to you about why we focus so much on genes, even in the circumstance that both genes and some environment are likely to be playing a role in the etiology or the cause of autism. Now, we know that genes play a major role, but we also know that this is not a simple genetic disorder like sickle cell anemia or Huntington disease. What that means is that there's not a single gene or a single change in a single gene that invariably results in autism. I'm going to give you very specific data about now how many genes we're estimating can contribute to the disorder. But this is like many of the disorders that are common in the clinic or in the emergency room, whether it's hypertension or cancer, diabetes, that what we see is that there's a genetic predisposition, but that how that manifests and whether or not it manifests from a given genetic change or genetic variation is probabilistic and not set in stone. Now, until very recently, and this is where the point about the recent revolution in autism genetics really is pertinent, is that for decades, my lab has been at work at this for 20 years, and until recently it's been extremely difficult to reliably identify and systematically identify autism genes. And that has presented fundamental challenges to our ability to think rationally about how we might treat the disorder. So we have very limited treatments, somatic treatments. The mainstay of treatment are behavioral modification, but the medications we use are really targeting behaviors, etc., that go along with autism often, but are not the core social features. That problem that we don't have medications that that are able to be helpful, and that our behavioral interventions are not as helpful as we'd like, to some degree as a consequence of just fundamentally not understanding what's going wrong in the brain. And so this bullet point here is that when we don't or have not had traction on what the molecular or cellular or brain circuit level mechanisms are that are going wrong, we can see when we look across other areas of medicine that having that kind of insight, whether it's understanding the molecular changes in cancer or in immunological disorders, that those kinds of understanding of the kind of basic processes that go into this or go into pathology have been critically important in driving fundamental change in our understanding and our treatment of those kinds of conditions. And I'm going to argue today that we're just at the beginning stages now in autism because of our ability to reliably identify molecular clues and molecular contributors to the disorder. So that brings me to the last bullet point on this slide, which is that there are many ways to think about studying autism, and all of them are tremendously important. My lab has focused on one area, which is to try to use gene discovery as one way in to kind of get a piece of string that we can pull on to begin to unravel some of these questions about the underlying causes of autism that may lead us to better options for treatment and intervention. So when I started 20 years ago, and actually until recently, I think there were a lot of people who felt that this was potentially a fool's errand, and that's not unreasonable. I've got a picture here of DNA. And the human genome has 3 billion bits of information in it that's carried in every cell in the body that has a nucleus. And really what we're looking for with regard to autism, we ask the question about what genes are involved, is looking at a very, very tiny fraction of those 3 billion bits of information, maybe just looking for a single bit of information that may be abnormal and leading to the developmental abnormalities. So there's no question that that's a needle in a haystack, but when you compare that to how complex the entire rest of the problem is, so there are 3 billion bits of information in the human genome comprised of 4 constituent parts, abbreviated A, C, G, and T. You compare that to the number of cells in the human central nervous system, which is 100 billion. If you want to understand the connections in the human brain, there are 100 trillion connections in human brain. And so consequently, one of the reasons for the strategy that I'm describing for you today is that in some respects, looking at the genetics is the simplest part of the problem from a biological standpoint. Now, ultimately, as I said, what I hope is that this leads us to be able to traverse from the 3 billion bits of information in the human genome all the way to understanding how to develop novel treatments. But one could ask the question, is it really plausible that a small change out of 3 billion bits of information in the human genome really could reliably lead to predictable changes in social behavior in humans? And I'm going to give you some evidence later in this talk that that is in fact the case. Before I go on, I feel like I want to make sure that we're all on the same page, and so I'm going to just give you a little bit of genetics 101 to make sure that, or at least to hope that the rest of the talk is entirely comprehensible. So what I want to focus on is that I've already told you that the human genome has 3 billion bases. What I think people often fail to appreciate is that if you take any 2 individuals on the planet and you look across those 3 billion bases, we are 99% genetically identical to each other. Now, it's extraordinarily interesting. Obviously, it is data that suggests that we're much more alike than we are different. At the same time, when you're thinking about what we're engaged in the lab, which is to try to identify disease risk, that also simplifies our problem a bit. Because from my standpoint, as a geneticist interested in gene discovery, I'm not worried about the 99% of the genome that is the same across individuals. That part I don't need to worry about. What I need to worry about is where individuals are different in their genome. Because to the extent that a disorder is a consequence of genetic risk, or there's an increased liability due to genetic risk, it's not going to be in the part of the genome that is similar across individuals. It's going to be in the variation in the genome that is present in the subset of people who carry that disorder. So we really are focusing not on genetics and genomics as a whole, but really on the idea of genetic variation. And I'm going to spend a lot of time talking about looking for variation in the genome that is contributing to the disorder. In order to do that, I'm going to give you some broad categories about how to think about genetic variation. So one is that most of the variation in the genome actually is quite common in the population. We define it as more than 1%, but there are variations in the genome that half of the people on this call might have in their genome. And most of the variation in the human genome has nothing to do with disease risk, but has to do with who your ancestors are. So we call that variation common variation, and that's probably the last that you're going to hear about common variation on this talk, because what we're going to focus on is the inverse of that, which is variation that is rare in the population, so present in less than 1% of the population. Now, the other thing is that variation, often we think about genetics as being synonymous with transmission from one generation to the other. But in fact, most of the genetic variation in the genome is passed from grandfather to mother to daughter, etc. But there is a small amount, actually quite a small amount of variation that arises new in each generation. And this is a natural process. It helps drive human diversity, but it's at a very low level. So when we look across the genome, there may be, out of 1 billion bases in the human genome, across that entire genome, there may be only 100 new mutations per generation. So we call those mutations de novo. The last bit of lingo that I'm going to give you is that I've been talking about the human genome, but what I want to focus in on for much of the rest of the talk are genes. And the reason I'm doing this is, while it only constitutes about 1% of those 3 billion bases, it's the part of the genome that we understand the best. We understand how the letters in the genetic alphabet within these units called genes code for proteins, which are biological machines that drive forward biological processes. So while there are 3 billion bases in the human genome, there are about 21,000 genes in the human genome. And it was a big surprise at the time that the human genome was first sequenced that that number was so low, because mice have about the same number. And most bets were that humans would have at least 4 times as many genes as mice. It turns out that we have almost the same number. So I've told you that most variation in the human genome is common, but I've also told you that I'm going to focus on rare variation. And there are a couple reasons for that. So one is that rare variation, when it happens and it's related to a disorder, often carries very large risks. And I don't have time to go into great detail, but we think that that's a consequence really of natural selection. If there's a rare mutation in the genome that has a significant effect on something like autism or some other developmental disorder, it will disappear from the genome over a short number or it will be driven down in frequency over a short number of generations. So when there is particularly a new mutation in the genome, it often is something, if it's related to disease risk, that has a large effect. And I'll explain as we go along why we felt like that was a place that we wanted to take a look. And then the last bit of Genetics 101 that I'm going to give you is that I've told you about the genome, I've told you about genetic variation, I've told you about genes. This process of genes being turned into proteins, I just want to point out as something that's highly dynamic, highly regulated across development, and can be very specific to an individual cell. So actually the tremendous complexity in the human organism, what we found is that it really is not so much a consequence of the fact that there are 21,000 genes, but it really is how those genes are orchestrated and the diversity of protein products that are created in human that separates us from other organisms like the mouse. All right, so I've told you that about sort of characteristics of the human genome and genetic variation. And the last bit of news that I'm going to give you is that that variation in the genome can either involve the structure of chromosomes, which are the scaffolding that carries the genetic material from generation to generation, or it can be in the sequence of the DNA, and that is represented here on the slide. On the left, what it's showing you is a diagram we call an ideogram of a chromosome, and you can see in the section B that the middle chromosomes are a representation of what would typically be expected, what we call the reference genome. And what we found is that while it used to be thought that we all were walking around essentially with normal complement of chromosomes, and that if you saw a change in the structure of a chromosome, that was automatically something wrong. It turns out that, in fact, there's normal variation in all of us, both in the structure and in the sequence, so that many people at a given region of the genome might look like the diagrams on my left, where they might not have a section C and go straight from B to D, or on my right, they may have an extra section of section C called, and we would call that a duplication. So that's the variation in chromosome structure, and then on the right is variation in the sequence of the DNA. You can see that I've shown you a chromosome, unraveled the DNA, and then you can see those 4 constituent parts, A, C, G, and T. There are 3 billion of them in the human genome, and then we're pointing to just one change in the letter code that could lead to a change in a protein that could lead to a genetic risk for a disorder. So in fact, we are looking for a needle in a haystack. There's no question. In fact, I think people have done the calculation, and it's much worse than looking for a needle in a haystack. There are far fewer than 3 billion bits of hay in the stack, the one that I'm showing you. But there are really 3 main reasons that now we've been successful in being able to identify the needle or needles in this haystack. So the first is really just simply a consequence of technology, both computing power, which has really advanced, as everyone knows, very dramatically over the last 20 years, but also just in our ability to actually take a look at the genome. A lot of the reason that it was so difficult previously is that we actually couldn't get data directly from the genome. We would take 400 data points from the 3 billion in the human genome and then try to infer what was going on. And now there's technology that allows us to actually read the sequence of the DNA or look at very high resolution for those changes in chromosomal structure. And that's been absolutely transformative in terms of our search for the needle in the haystack. The second really has been a cultural societal push, which is that what we found is that in order to be successful in searching for the needle is we need very large groups of individuals who are willing, both patients and families, to contribute. And there really has been a dedicated effort over the last 20 years, not only by advocacy groups and funding groups like the Brain and Behavior Research Foundation to help promote research, but then also a real push from those kinds of organizations and family organizations to make sure that scientists are sharing the data. And again, sharing the data because that dramatically increases the power of the experiments. So you put together the technology and the advocacy and philanthropy, it has really been tremendously powerful in changing the game for us. And then the last bit is really scientific and a bit of luck, is that remember I told you that there are very small numbers of new mutation every generation in the genome. It turns out that we began to have evidence about 10 years ago that new mutation might play a role in autism. And that has turned out just to be a lucky break. And the reason that it's lucky is because if we were having to sort through 20,000 or 100,000 or 1 million genetic variants in every person and trying to figure out which one might be related to autism, you run into a very serious signal-to-noise problem. I've told you that across the genome there may be only 100 new mutations per generation. And when we look just at the genes, the 1% of the genome that we're most interested in, we get at typically only one of these mutations per generation. So what that means is that when we're looking through these samples of patients to see what's different about kids or families with autism, we're looking at really very small numbers of observations and where differences can be really magnified. You're not talking about a change of 1 out of 20,000 or 100,000. You're talking about, 2 instead of 1, and that's much easier for us to identify. All right. So I'm going to tell you about a series of experiments that are all going to rely on something called the Simon Simplex collection. And this collection actually was built based on the initial clues that we were having that de novo mutation may be playing a role in autism. So Jim Simons, a philanthropist in New York, was aware of some compelling early evidence that de novo mutation would play a role, but realized that there weren't many samples in which those kind of mutations could be easily identified. And he set out with a number of scientists on the advisory board there to build a collection that would help solve that problem. And this is the way that they did it. If you look at this family diagram, it's representing that in each of the families in the Simon Simplex collection, there could be only one affected offspring. And everyone else in the family who was available was questioned or given a clinical battery in order to determine whether or not they had evidence of autism, and there could not be another first-degree relative who had autism. And the reason that they did this was that if you think about a de novo mutation arising spontaneously in a new generation, that is like a genetic lightning strike. And so they were looking for families that look like they were hit by lightning, by genetic lightning. And it also gives us 2 very simple things to do in order to build this experiment. The first is just with whatever assay we have to compare the parents to the children, anything that's present in the children, any variation in the genome that's present in the children that is not present in either the mother or father is a new mutation or a mistake, and we can easily tell the difference between the 2. And then what it also allowed us to do was to look for new mutation in the affected child, but then have the perfect control, which was to look in their unaffected sibling. And so we could ask about, is there a greater rate of de novo mutation in the affected versus the unaffected child in the same family? All right. So this is sort of now going to track you through leveraging this sample, which ended up being 3,000 families that were collected across 13 sites in order to build the Simon Simplex collection. The first thing that we were able to do was back at around 2010, so now 5 years ago, we weren't able to read every letter of the DNA code. We were able, because of some new technology, to look at high resolution at changes in chromosomal structure that I showed you previously. And so we looked at high resolution in the Simons families to see whether or not we could find an increased rate of new structural mutation in kids with autism. And we found that, in fact, it was the case. This was a replication of work that had been done several years earlier that had led to the formation of the Simon Simplex collection. But now we were looking, instead of at 100 families, we were looking at that point at 1,000 families. So it was a very strong replication of that initial result. This is Stephen Sanders on the left, who led this in my lab, and it was published in Neuron in 2011. And what I want to focus on is we were able to look across the genome. We were able to find that there was an increased rate of new mutation in kids with autism, structural mutation. And we were able to use that information in order to narrow in on a couple regions of the genome that were repeatedly unusual in kids with autism versus controls, and provide very strong statistical evidence that that was related to autism. I want to focus you on just one of those segments, on chromosome 7. This is a small segment of the genome that contains about 25 out of those 21,000. I'm going to show you a video now just of typical autism, a child with duplication of chromosomes. This is what that would look like for typical autism. And there's no sound here, and you don't need it, I don't think. So I'm going to click and show you this. So the child is not speaking. It says father on the right, an examiner on the left, and everyone who's shown in videos here is given permission to be used for educational purposes. And I think it takes about 3 seconds into the video to see the kinds of repetitive behaviors that we were talking about. There was hand flapping, which is classic for autism. But I think you can also get a sense very quickly that for this child who is, just reaching adolescence, that the relatedness that that child has socially with the people in the room is unusual and is really what we're talking about as a core feature of autism. So I'm showing you this, but now what I want to do is to contrast that. This next video is going to have some sound, and I'm gonna move my phone over so that I hope that you all will be able to hear this well. This is the exact same region of chromosome 7. Nothing different about it. In this case, instead of having 1 too many copies, there is 1 too few copies. And every child that you're going to see in this room has lost that exact same portion of chromosome 7 that increases risk for autism, as I just showed you. So here we go. I hope you all can hear.
Nice to meet you, bud. Nice to meet you. Great. Great. Got great hands. Hello, buddy. How are you? I'm really well, thanks. Oh, thank you. Thank you. What's your favorite color and what's your favorite TV show?
Kids love to ask questions, but not as much as these kids. My favorite color is blue. I have met Barney the dinosaur.
Yeah. Where do you live?
I live in New York City.
Do you have any sons or daughters?
I do.
Wow.
I have 2 daughters and a son.
Wow.
And kids love to make friends, but not like these kids.
What nationality are you?
I'm Italian. Oh.
How are you, buddy?
So I hope you were all able to hear that. Even if you weren't, I hope that you could see just by looking at the kids in the room who all have this loss on chromosome 7, how intensely interested they were in the interviewers, Chris Cuomo from ABC's 2020. This is absolutely characteristic of this. This is, by the way, called Williams syndrome, and it is a highly reliable, what we call behavioral phenotype, or set of behaviors that goes along with having this loss of chromosome 7, where there's an intense interest in reciprocal social interaction. So I wanted to show you this in part because I think it's a beautiful experiment to infer that this long trek that we proposed at the start 20 years ago in the lab, or that I proposed at the start of this talk, that somehow we could get from a small change in the human genome through the 100 billion cells, through the 100 trillion synapses, and know that actually what we were finding at the molecular level could have a profound impact on something as complicated as human affiliation or social behavior. And I think that there are many examples, but I think that this is an excellent one, that in fact it is not a fool's errand, that we can see that a change in only 25 out of 21,000 genes can reliably be a rheostat of social behavior. There are arguments about how opposite autism is from Williams syndrome. I don't think it's really important. I think what's important is that you can clearly see that by changing the number of copies of this small section of the genome, that you can have a profound influence on how people interact and how interested, how able they are to have reciprocal social communication. So this was, an exciting finding. I think it helped drive us forward further, but it really is not the experiment that we wanted to do. And the reason is that 25 genes sounds like a small number, but if we really want to use that information to begin to understand pathophysiology, we really want to understand what the molecules and cells are doing, 25 is too big a number. What you really want to do is you want to be able to go to your neurobiologist colleagues and say, it's this gene, it's this mutation, though it's rare, it's having a large effect, and allow them to study that at the bench. And up until a few years ago, that was really not possible. So as I said, by 2010, we could look at chunks of DNA that were somewhere in the neighborhood of 50,000 bits of information, but we couldn't get down to a single, what we call single base resolution, being able to look at those constituent parts of DNA, A, C, G, and T. And that has all changed dramatically as well. And I'm going to show you, give you a nice example. I'm in Silicon Valley, and so I use this often in my talks here. This is a graph of the cost of sequencing all of the genes in the human genome for just over really the last 15 years, and it stops at 2012. The green line is the cost of sequencing all the genes in the genome. Actually, no, this is sequencing all of the genome, I'm sorry, so all 3 billion bits of information. And what you can see is that the line on top is Moore's law, which people, it is what accounts for the tremendous revolution in computing power. And so you can just take a look at how profound the drop in cost has been in allowing us to sequence. So what this has enabled us to do now essentially already is to read every letter in every gene in every person that we're looking at in the Simon Simplex collection. And actually now it costs about $1,500 to sequence every one of the 3 billion bases in every person that we're studying. And so now the Simon Simplex collection, we're moving on to do what we call whole genome sequencing. But I'm going to show you the experiments that have already been published looking at individual bases of DNA just in the 1% of the genome that codes for genes, which we call the exome, starting in about 2012, our lab and several others. You've got papers here from, the second one down is from Broad and MIT, third is from University of Washington, the others are from my lab, and what they're all showing is the same answer. There is an excess of de novo new mutations in kids with autism. There's a particular excess in de novo mutations that kill the protein, so they're changing the letter code in a way that's having a very deleterious effect on what the biological process should be of taking that gene and turning it into a protein. And so those were papers that all came out, all got essentially the same answer in 2012. And then there were 2 papers just this past November from large-scale consortia, one called the Autism Sequencing Consortium, which Dr. Bornstein already mentioned, and then others from a group of labs collaborating around the Simon Simplex collection, showing the same thing. So, and I always have to show the folks in the lab that do the hard work. That's Stephan Sanders that I mentioned. Jeremy Wilsey was a graduate student at the time. And then Sean Dong on the right, you can see her name down on the bottom article on the left, who was a graduate student at the time, shared between us and Beijing University. What did we find? So we were able to use this approach. We found 9 genes that everyone agrees are contributing to autism, just in the way we were able to find those regions of chromosome 7 that were repeatedly showed new mutation in kids with autism. We were able to find 9 genes in which there were enough observations among unrelated kids in the same gene that we're confident that those are autism genes. And by 2014, we've been able to identify 51 more. We know now that new mutations contribute to at least about 20% and maybe 30% of cases of autism. I'm going to pause and say that's still a minority. We know it's a minority. But as I told you at the beginning, the strategy here is not gene discovery as an end to itself, but gene discovery as a way to begin to help lead us to biology, and that then is the reason for the next bullet, which is while these mutations are rare, they carry very large biological effects, and that's really good for our neurobiologist colleagues who want to understand what this is doing at the cellular, molecular, and circuit level. All of the groups that I've told you about simultaneously found that most of these types of mutations, again, this is about contributing to about 20% as far as we can tell at this point of cases of autism, that most of these de novo mutations at a single letter in the DNA code were coming from father's sperm as opposed to from mother's, and that the risk for these mutations arising increase with age. Now, I have to point out that this is an interesting observation, and it gives us, I think, some insight down the line to begin to think about what environmental factors besides being male or older might increase the rate of de novo mutation. It accounts for a very small percentage of the purported increase in autism prevalence, but we definitely see that there's an increased risk of de novo mutation in older fathers. We're able to predict now that there are 400 to 1,000 genes contributing to autism via this mechanism. And we're also able to find, and this has been sort of mind-bending for the field, I think, in a way, is that while it's very reliable that these genes increase the risk for autism, some of the very same genes will increase the risk for epilepsy, but not autism in another child, schizophrenia, but not autism, intellectual disability, but not autism in another child. So what we're finding is that these genetic predispositions, which is what they really are, appear to some degree to set the stage for a range of neurodevelopmental disorders. And in some cases, as I said, these genes are not fate. Even if you have one of these mutations, you will not necessarily show any of these outcomes. And so those present some challenges for us. For those of you who have been following the literature, I've stuck in a slide with gene names to show from our group here at UCSF the genes and their level of statistical significance for association with autism using a false discovery rate metric here. I'll leave this on for a moment for the neurobiologists, if there are some on the call, to look and see. I think the main point here is that there are lots of genes and that they do many different things. So if you just run down the list and have a rough idea about what the function of these genes are, they range from being involved in synaptic functioning to the growth of neurons to specifying how other genes behave, etc. So it's that the first step in identifying genes has not led to a unifying biological theory of autism. But what it does do, I think, is lay the foundation for the next epic. And I'm going to just, the last 5 minutes of the talk, I'm going to try to give you an idea about how we're thinking now about moving ahead. We now know that we can reliably, you saw all of those papers all coming to the same answer. I have to say that in psychiatric genetics 5 years ago, that might have been unheard of, that we never found the same answer. But now, routinely, we are across many psychiatric disorders for the same reasons that I've described here. Large samples, data sharing, and profound advances in technology and methodology. One of the ways to think about, if you think these are all clues to autism, we know that they increase the risk for autism. We can quantify how much we have confidence in that. And when we can begin to ask of this set of genes, which could ultimately number between 400 and 1,000, can they begin to form a pattern or a picture? Now, one way to proceed would be to take each gene one at a time and try to see what it does, say in a mouse or some other model system, try to figure out what was going wrong. And I think that that can be very valuable, and there are people who are certainly picking the best ones among these and doing that. But I think it's also reasonable to think that if we know that all of these genes converge somehow on social disability in a highly reliable diagnosis that most psychiatrists can agree on when they see the child to a great degree, that there must be something that binds these together. So one way to think would be to ask, what do these genes do? Are there any cellular processes or molecular processes that are overrepresented simply by looking at this list? And in fact, one of the things we were able to do very quickly is to identify that there are a dramatic overrepresentation here of genes that are involved in regulating how other genes behave, and in particular how the chromosomal structure that contributes to that regulation is managed. And we call that chromatin modification, but all of the genes here that I've highlighted in a box, remember there are 21,000 genes in the genome. There are much, much, much smaller number of genes that are involved in chromatin modification. And we know that this is a statistical overrepresentation. Now, that's helpful, and people are going to start to work on chromatin modification to understand what that contributes to autism. But I think that there's another question that our group has been particularly interested in, and this is in collaboration with Nenad Sestin at Yale, who's a developmental neurobiologist. And we had the thought that, these are developmental disorders, so it might be important not only to ask what the genes are doing, but are there groups in which you can see that there's some convergence in space and time, that maybe there are vulnerable points in development in which different sorts of genes that might do different things could converge on an autism phenotype because they would disrupt some important convergent process in development. And so we set out to try to answer that question. Jeremy Wilsey on the left, who was a graduate student at the time again, who led this effort. And really what we did is we were able for the first time to have a set of genes where we had very strong mutations. We knew that they were relevant for autism. But then because of the work that Dr. Sestin had done, we also began to have a roadmap to how genes are turned on and turned off throughout brain development. A very painstaking set of experiments in which he took fresh postmortem brains, so right after death, across the lifespan, dissected out different brain regions, and then was able to look at the activity of those genes in each of those developmental epochs and time periods. So really this gave us a roadmap across brain development. And what we've done essentially is to start with these genes, and we can essentially, using his data, go back in time to very early development and then roll the tape forward to see whether or not the activities of these genes converge at any particular point in development, special point in development. And in fact, we were very surprised to find that just with our initial set of very high confidence genes, that we did find that as we rolled the tape forward, that there was a network of genes that were much more closely related in terms of their activity than we would have expected by chance. And also started to identify other genes that we were finding through other methods were related to autism risk. So essentially what we were able to do then was to take a look at that point of convergence in space and time and begin to place it in human brains. So we were able to determine that that network of genes was particularly relevant for the front part of the brain called the cerebral cortex. We were able to find that that network was really most relevant not for the entire cortex, but just for a subset of the layers that build that cortex. And then we were able to find that those layers were really, this work was really predominantly represented in a particular cell type, in a particular layer, in a particular brain region at a particular point in time, which was development. So using this approach, we were able to go from taking autism mutations, combining that with rapidly advancing understanding of what genes do in brain development, identify this network, narrow in on a brain region, on a time, on layers, and now to a cell type in which these divergent autism genes come together in order to cause pathology. So I hope that that gives you a sense of where we are and where we're headed. We've gone from not being able to identify in a reliable way. There were discoveries early on, but they were, sort of sporadic points of insight, as opposed to a reliable, reproducible, systematic way to identify genes, which we now have. And all the groups, whether they're in Washington or Massachusetts or San Francisco or back at Yale, are all being able to use the same method to increase the number of genes that we're identifying. There are simultaneous advances in basic neuroscience, one of which I gave you a nice example of, I hope you feel it was nice, that are providing us unprecedented opportunities to begin to interpret what these genes mean. Well, the process, I know, for every family that I see in clinic is too slow from the standpoint just of science, of what it is to be in the lab, that this is really a profound change in our understanding and lays the framework, we hope, for an even more important and profound change in our understanding of the neurobiology of autism, which, again, we can only hope will not only lead to a change in how we think about diagnosis and treating, but we also have a strong feeling that science and scientific insights into disorders like autism or childhood onset schizophrenia or schizophrenia will also begin to change societal views of mental illness and help reduce stigma, in addition to being able to directly provide better care for patients and their families. These are the folks that were involved in this work. It really takes a village, and in fact, each one of them has a lab full of people that I'm not listing here, but Nanad Sestner, I've already mentioned at Yale, Jim Noonan, who's a chromatin biologist at Yale, Bernie Devlin, Catherine Rader are statisticians who we desperately need in order to be able to interpret the mutations that we had. And some experiments I didn't show you were actually being able to use zebrafish to further interpret these mutations. And Antonio Heraldez on the right is involved in that. We have a great clinical group here now that have been recruited in who are leading the clinical effort and helping to ascertain additional patients, and I wanted to highlight them for you as well. This is our happy group. We always have a ton of fun in every lab meeting, and then to acknowledge the funders who have helped make this possible, Simons Foundation, NIMH, Overlook International Funds, and most importantly, as I said, without the families who participated in this and allowed us to look at 3,000 families with this, high-performance technology, we would have had nothing. So they are in every way what drives the research and the most important resource in moving ahead in this line of work. So thanks for your attention, and I think we still have about 15 minutes or so for questions.
Yes, we do. Matthew, what an excellent presentation on complicated subjects. It was wonderful how you sort of took us back to Genetics 101 and then used that to explain in really a very straightforward way these complicated issues.
Thanks.
One of the questions that we received a number of times from people relates to the rate that autism has been diagnosed and the increasing rate and concern. And I'm curious, where do you see this increasing rate? Is it better diagnosis? Is it that there's actually more cases? Is it a combination of the two? And how does it relate to some of the genetic work that you've been doing?
Great question. So I think the simple answer is that I think there's very good evidence that it's both. I think that there are studies that, there's no question that the diagnostic criteria have changed and continue to change, and that even beyond criteria, that as people get more experience and become more aware of these syndromes, even outside of sort of formal clinical settings, that leads to an increased ascertainment within the population. So there's no question that a significant portion of the increase in the population is a consequence of ascertainment, changing definitions, et cetera, better detection, appropriately better detection. There were many kids who could really use help and support who were not being identified previously. Now, at the same time, there is evidence from epidemiological studies that even when you factor all of those things in, that it looks like there's an increase, a much smaller increase than you'll hear on public service announcements, et cetera, but somewhere in the neighborhood of a 3- to 4-fold increase in risk over the last 40 years or so. And that's not inconsequential from a genetic standpoint or from any standpoint, right? So the question of how that could be happening. I think, I talked about the fact that this strategy really isn't about explaining everything. It's about getting a foothold. And in fact, I would argue strongly that because we understand the genome and it's a finite set, that it's not only easier than studying the 100 billion cells and 100 trillion synapses, but it's also easier than, at this point, than studying all of the myriad possible environmental influences that might be playing a role. We know that environment plays a role in autism risk. So there's no question that could be contributing or is contributing to some degree. And so one of the things I think that our research shows is that by understanding one side of the coin, you can begin to understand the other a bit better. We know now that de novo mutation is going to account for a percentage of patients, and we can begin to ask questions in a much more kind of detailed and specific way about what kinds of things we will have better hypotheses and then better ways of measuring. Does this environmental factor increase, for instance, the rate of de novo mutation in parents in a way that could begin to account for some of the difference. But we will also, as we better understand genetic risk, I think it will help us when we understand the underlying biology to come up with better hypotheses about what it might be in the environment. And the last thing that I always say when I take this question is that one of the things that, there's so much that we don't know. What we do know is that there's no association between vaccines and any purported increase in risk in autism. And there is no credible evidence that that's what's going on. Doesn't mean that environment is not important. It just means that we've looked hard at that question, and the answer has been repeatedly that they are not playing a measurable role in increasing the rate of autism.
I'm glad you brought up that issue of vaccines, because that was the question that I was going to ask about to clarify that. I'd like you to speak a little bit more about the issue of people who may have some of these de novo genetic risks, but then ultimately don't develop autism.
Right.
Any sense as to why someone does and someone else doesn't?
It is really the key question, and in fact there have been some really wonderful examples lately of people being able to identify protective factors, genetic protective factors that are really a direct avenue to thinking about therapeutics. Because if we can understand why, even in the face of a significant risk, someone is not getting a disorder, they're the exception. But if we can figure out why that happens, that would be a tremendous biological insight. There's some things that we already know that we're trying to sort out. So I didn't talk about it much, but it's absolutely the case from all of our data now that we can get very granular about the contribution of de novo mutation. Girls are protected. You need to have a bigger genetic hit in a girl in order to have to see evidence of autism. We can see that on a population level. So there's something about the female brain or female development or some risk in males that is relevant. And I think that's been an observation for a long time, that there's about a 4-to-1 excess of males, roughly. I mean, it depends on how you make the diagnosis, but it's a reasonable number. That's a dramatic overrepresentation of boys. So we know that boys have been vulnerable, but we are having mounting evidence that girls are in fact protected. And so there, I think it's going to be a very exciting area of inquiry to try to sort out why that's the case, and to see whether or not we can leverage that for therapeutics development.
The issue of other neurodevelopmental illnesses for people who might be at risk. Could you say a little bit more about that? Why would somebody not have autism but then develop childhood schizophrenia or a different disorder?
Again, these are, the work that we're doing is now laying a foundation. It's helped to when I say we, I mean that as a scientific community, not our lab, that is really laying the foundation. And every question that you're asking not only is like a wonderful question, but it also is something that I think for the first time we have some possibility of, or began to have ideas about how we could think about designing experiments to begin to answer those questions, because we can hold one thing constant, because we can know, for instance, we could get everyone who has a risk factor, and in fact the Simons Foundation is doing this around one of the structural risk factors in autism. Well, it's a structural risk factor for neurodevelopmental disorders on chromosome 16, and they've gone around the country and they're trying to find, regardless of diagnosis, who's got this, and then can we move forward through development and see what happens, and try to begin to leverage that to understand why, not only in some cases does it lead to nothing, but how it might be different and lead to these other disorders. I think again, if we knew that, it would be a tremendous insight into the biology of these conditions. I think in my mind the analogy is the following. We're identifying risk factors that are impinging upon fundamental processes in brain development, when that happens, there's not there are. Then you can think about, I'm going to mix metaphors here, but it's like dropping a pebble in the water, and then there are ripples. And the fact is that the way that then development transpires and all of the other things that might go in to dictating what an outcome might be has some possibility of playing out over developmental time, which is one of the reasons we were so interested in maybe what it is that defines autism is not exactly what that gene is doing, but when and where it's doing it. So you could have one hypothesis that the point of vulnerability in schizophrenia, that maybe the same process is deranged, but the point of vulnerability may be later in life because we don't see onset, frankly, typically until the third decade. So. So I think the consensus is sort of around the idea that these risks are not specific to an individual disorder, that they're disrupting normal development, and then the key question is going to be, how are they canalized? Why does this kind of general risk head in one direction versus another through development? And again, just like the protective factors, if we knew that, that would be a profound insight. Having specific genes, being able to look at everyone in the country with, where we know that this section on chromosome 16 is relevant, begins to give us some hope of answering those kinds of very complicated questions.
For really a large number of people who are listening to this webinar right now who have a child with this condition,
yeah.
What do you say to them?
So a couple things. One is that I've been a clinician for 20 years as well as working in the lab, and I think it's really important that scientists recognize that while we think that the progress is tremendous and exciting, that for every family that comes in that we are moving too slowly. And I think from both sides that acknowledging that and having a dialogue about that is really important. I think I can talk more specifically about what, genetic testing, et cetera, but I think the underlying idea about partnering between families and people who are doing basic science research is so important, and we haven't done ourselves a favor in genetics by overpromising early on and not delivering. I think if there's one message that I could give to families right now, it's that there really has been a profound change. It's not just in autism, it's in schizophrenia, bipolar disorder, and a host of other diseases for which now we're able to think about precision medicine. But I hope that that we can continue to partner because, as I said at the end of my talk, without the contribution of families who have children or adults in their families who are affected and who are contributing to our studies, we've got nothing. So I think that's very important. From the standpoint of the actual clinical relevance right now, I think that there are some potential benefits, mostly in terms of thinking about things like, I'd say one is just the intangible feeling like you have some idea what's going on. I think, we may not be able to say, we have a special treatment for you because we know that you have this mutation, but I do think that there is some value. Certainly some families would really love to know whether or not there's something that they can, understand in a much more concrete way so that we're not just telling them we have no idea why this happened and we don't know what's going on. So I think that can be valuable. I think as this is going to start to become more commonplace to have genetic testing in the clinic, and I think as it becomes more commonplace and more data is collected around things like developmental course, how people respond to different treatments, we're just starting to do that. But I think that there will be future value, not in the distant future, but the relatively near future. So if you know that you have a particular mutation, that being able to track, other families or other kids to get a sense about how the research is proceeding and whether or not there's any new clinical information that might be relevant is going to become increasingly important. Right now, it's mostly a research issue in terms of, we can say that there's a gene, but there's so far nothing that says, we know that this treatment will work. But there are genes, for instance, we already know that Fragile X mental retardation protein, that there's enough known around the biology there, that there are ways to start thinking about doing clinical trials that would target folks with particular mutations. So I do think that, as I said, particularly around this partnership between families and researchers, that having the families know about the genetic situation can help contribute to a variety of studies, not just genetic studies, that are going to begin to take a look at neuroimaging or treatment response, et cetera, based on these genetic findings.
As our time runs out, I want to say, Matthew, thank you so much for an extraordinary presentation, and more importantly, for the work that you've done all these years, the work that you're going to do, and for the passion that you have about this, which comes across so strongly. So thank you very much.
Thank you so much, and thanks everyone on the webinar. I appreciate your attention. Bye-bye.
I also want to thank all of the people who have joined us today. All of the research we fund is made possible through private donations. So if you'd like to make a gift, please visit bbrfoundation.org or call 1-800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it with a family member or friend, visit the webinar page on our website. And I hope you'll join us next month when Dr. Nina Schooler, Professor of Psychiatry and Behavioral Sciences at the State University of New York Downstate Medical Center, will present a webinar entitled Early Team-Based Treatment for People with Psychotic Symptoms. This will take place on Tuesday, April 14th at 2 p.m. Eastern Time. Once again, thank you for joining us today, and have a nice day. Take care.